All right, so now let's take a look at some checkpoints or some controls of the cell cycle. Again, how do we tell cells when to divide and when not to divide? Well, we have to have some control over when it divides and when it doesn't. And we don't want it to divide when it's not ready. All right, so we want to have some checkpoints. So you can see here, I'm going to go over these in a little more detail. So after G1, there has to be a checkpoint that says, are enzymes ready to go into, can DNA synthesis begin, right? So there's this checkpoint. Do we have all these enzymes? Do we have things ready to go before we go and divide the DNA, right? So there is, you're going to see here, there's something called cyclin. I'm going to show you that here on the next slide, so don't worry about it too much right now. And then it'll go through and divide if it works. And then we have G2. Before we go through mitosis and divide the DNA, has DNA synthesis, did we divide the DNA okay? Did we copy it okay? Right? So let's make sure before we go and split this into two cells, let's make sure that we copied the DNA okay, that everything's fine. All right, and now we're like, okay, yes, everything's good. Now we can go to mitosis. And then during mitosis, are all the chromosomes lined up correctly before we divide that and before we split into two new cells? So basically what I'm saying is there's these checkpoints along the way that control whether a cell signals it whether to divide or not. And if it's not ready, again, apoptosis can say, shut this down. We don't want this cell dividing and passing on damaged DNA or passing on the wrong thing to the next cell. All right, so we can stop it at that point. All right, so next is, these are a type of cell checkpoints that I want you to know. All right, so we have this thing called, it's a protein called CDK. All right, and this is called cyclin dependent kinase. All right, so there's that word kinase again. Again, it becomes activated when you add something to it, especially a phosphate. Right? We're not going to see that as much here, but this is a protein that's present during G1. It's present during S and G2. Right? So you can see CDK is present basically during all of interphase. Okay, we have this CDK. And there's some checkpoints there, like before it will synthesize DNA, is this CDK available? All right, so that's sort of a checkpoint before you go into this copying the DNA phase. Is this CDK available? Okay, now, as we get to G2 and as we're getting ready for mitosis, all right, we're getting ready to split that DNA. How do we signal that to occur? Well, now we have another protein called cyclin, and notice what it does is it bonds to the CDK. So it's got this little shape that will fit with the CDK, and you can see that when that does that, they fit together. They fit together, and now we have this thing called MPF. All right, so MPF, I'll put it over here. Oops, sorry. MPF stands for mitosis promoting factor. So notice, CDK is always there. When we're getting ready, the checkpoint here at G2, let's check, did DNA divide correctly? All right, if so, cyclin will bond to CDK, and when that does, it produces mitosis promoting factor that then signals, yes, we can go through mitosis and we can break apart this DNA. Okay? We can split it into two, not break it apart, like kill it, but to split it into two so that it can go to two new cells. So then during mitosis, this cyclin will get degraded because now we've said, yes, we can go in. And then during mitosis, the cyclin will be degraded so that we can then just have CDK left. And then the new cell will go through, right? So here is a signal when cyclin bonds to CDK. We get mitosis promoting factor, which does just that. It says, yes, it's okay to go through mitosis and to split the DNA into two cells and split the cells into two, right? And to copy those cells. All right, and then during that, cyclin will just be degraded so that we still have CDK. And then when a new cell is ready, the cyclin will be added and it will say signal that mitosis. So here's a signal that happens that tells a cell, yes, we're good to divide. Okay, now, Obviously, we know that some things can happen here that causes cells to divide when they shouldn't. This is what causes a lot of cancers. Cells are dividing when they shouldn't. So we get a signal somewhere that says, yes, it's time to divide, even though they should not. And that's what a tumor is. It's extra cells that should not be there. Okay, so to give you some examples of that. Let me go here. All right, so I'm going to go back and then I'll come here. So there are some outside signals that can occur. I'll do this first. So here's cells that are in like a little tray. Look, there's room to divide. So now here's that paracrine signaling and autocrine signaling. That will send signals to the other cells that, yeah, there's room to divide, so I can divide, I can copy that. But now look at this. The cells fill up this little tray, so they're in contact with one another, and they're sending signals to one another. There's no more room to divide. Stop dividing, right? So then they don't divide, right? It tells them to stop dividing. And then if I take some cells out, they will signal, yep, there's more room, it's time to divide. So sometimes that signal can be from that close contact, right? That juxtacrine signal can go through and say, don't divide, we're too tight with each other, we're space, yes, it's time to divide. So sometimes the signals can come from other cells, okay? And then sometimes the signals are from within. So I'm going to go through this a little bit at a time. All right, so there are growth factors, and these are like ligands that tell a cell whether to divide, and they're caused by certain proteins that are formed from certain genes, certain DNA in our body. So we have this thing called a proto-oncogene. All right, so onco refers to cancerous. An oncologist is somebody that studies cancer. So we have this proto-oncogene. So it's a gene that makes a protein. All right, so this proto-oncogene will activate cell division. All right, so its job is to signal a cell to divide. Okay, so it is this signal. It's the ligand that tells a cell to divide. All right, so these are called growth factor genes, right? It's telling a cell, yes, it's time to grow, it's time to divide. Okay? So these can become oncogenes or cancer-causing when they're mutated. And what that's going to do is it's going to, these are only activated when a cell needs to divide. But if they're permanently on, you're going to see that these cells, right, if they're permanently on, they're going to activate a cell to continue to divide. There's no turning them off. So these cells can become oncogenes if they are switched on. They are causing constant cell division. So these are sort of normally off until we need to divide, then they're turned on. But if they're always on, they're always telling cells it's time to divide, and we get these cells that are dividing when they shouldn't be dividing, right? And so that turns it into an oncogene. And this activates that cyclin that we just talked about, right? So that cyclin, remember, will bond to the CDK, telling a cell to divide. All right, so that's the way that this works. It's a signal that activates cyclin permanently. Remember, it degrades. It doesn't degrade. It stays bonded to that CDK, and it's constantly telling the cell to divide. Okay, so proto-oncogenes should be only signaling at some times for the cell to divide, and when they're mutated, it turns them on permanently, and cells will continuously divide. All right, and it becomes an oncogene, causing cancer, causing, when they're mutated, causing continuous replication of cells. Okay, we have another one called a tumor suppressor gene. This one is usually on, and it's stopping cells from dividing when they shouldn't be dividing, right? So that makes sense. It suppresses the ability of cells to continue to divide. So that's a good thing. We don't want some cells to continue to divide. So this one is normally inhibiting cell division. It's normally on, saying, hey, liver cells, I don't need you to divide right now. All right, and then you can tell when this is, if they're always on, when it's switched off in this case, it's going to stop suppressing the tumor, the growth, and it's going to cause the cells to divide. So this one is a little bit different if it's switched off. So this one switches on, it causes cells to divide. This one, if it's switched off, can cause cells to divide and cause tumors and cause cancer. All right, and an example of this is a type of protein called P53. All cancer has to turn off P53 because it will then, this normally inhibits cell division. All cancers will turn on P53 or switch, yeah, and it will basically, it halts that. You're going to see, I'm going to show you P53 here. All right, so all cancers have to shut down P53 activity. That's what I wanted to say. Okay, so I'm going to show you what, don't worry about this stuff because I'm going to show you what it does here. But P53 is an example of a tumor suppressor gene. All cancers will have to shut down P53 activity. And I'll give you an example since my husband's dealing with this right now. I know we'd had a DNA analysis, and his 17th chromosome has a deletion on the end of the 17th chromosome. What's cool is that I understand this. On the 17th chromosome is where this gene is, where P53 is made. His is deleted, so he can't make P53 to inhibit cell division, and his cells divide. All right, that is one of the reasons that caused his type of cancer, is that he has a deletion on a gene that should be making these tumor suppressor genes, and they don't. So it's telling his cells to divide when they shouldn't. Some of those bone marrow cells, all right, are dividing when they shouldn't. All right, so how does P53 work? All right, so this shows you a good graphic. So let's say there's damage to DNA, right? It could be radiation, could be multiple things that damage DNA. P53's job is to recognize that and trigger enzymes that will help repair that. Okay, so here's a repair. All right, so look here, this cell will divide normally because P53 said it's okay to divide. But here, P53 says, whoa, there's damaged DNA. I don't want this cell to divide. It triggers apoptosis. And again, it suppresses, if it notices that there's damage, if P53 says there's no damage, you're good to divide. But if there is damage, P53 should recognize that. It's a tumor suppressor gene, and it should cause apoptosis to stop that cell from dividing. That's what should be happening with P53. It's a regulatory protein that recognizes no repair, we're good to go, divide. No damage. If there's damage, shut it down. We don't want that cell dividing. That's the way it should work. With abnormal P53, or you're not making it, all right, so what happens is there's damage, and it doesn't recognize here, and it allows those cells to divide, right? It's turned off. So those damaged cells will continue to divide and divide and divide, and that's how cancer can grow and go to other areas, because it's telling this damaged DNA to continue to divide. Okay, and we don't want that. P53 is a tumor suppressor gene. It should shut that down if it sees damage. But it's turned off or not made, and cells will divide, all right, and pass on that damaged DNA again and again and again, all right? And that's how it can grow to other areas and cause other areas to start growing, and that's how tumors can spread, okay? So all cancers have to shut down. They have abnormal P53 in some form, in some way, right? That they shut that down. And we're not exactly sure why that happens. Some cases we know. There's a deletion, they're not making P53. That's why it doesn't work, because we can't make it. In other cases, we're not exactly sure what triggers that to not work. Okay? So that's P53. It's an example of a tumor suppressor gene. Okay? And I already went over apoptosis. Okay? So that is a way to control and to signal a cell when to divide. Is it okay to divide? Is it not okay to divide? There's checkpoints along the way that should work, and when they don't work, we cause a proliferation of cells, and that's what a tumor is. Okay? All right, that's the control of the cell cycle.